This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 822, recorded on October 26th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 50 degrees and it's sunny. There's not very many clouds in the sky. It's our one sunny day between the rainstorms. So I'm looking up what 50 is in Celsius. I'm getting it from the Norwegian weather app. 9 degrees.
Chilly. We were at 15°C and cloudy. It rained all night. And this morning, completely messed up the trains. I was talking to people on the platform because everyone's train is late, so everyone's talking to each other. We never talk to each other except when there's what's the word? Not diversity.
Diversion?
Not diversion. Something going bad, right? What's the D word for that? I can't remember.
Disaster
or something like that. And there's one lady. She left home at 6 a.m., got almost to New York, and then turned around and came back and was trying to get home because she didn't want to risk it. It was a really bad morning. The tracks get flooded. But I was telling her, I drove for 39 years, and now I take a train, and I love taking the train, but it screws up. However, the roads were really bad this morning, so I was glad that I didn't take the highway. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's overcast, rainy, dreary, dark, 57 Fahrenheit, 14°C. Been raining for days.
You don't have to commute anywhere.
No. Well, I mean, I have to drive my daughter around, but
yeah, that's nice.
Pretty soon she'll be driving herself around, and that'll be a whole different thing.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It's 58 and pretty gray. Obviously the same as what everyone else is having. And I'm very glad to be indoors today.
You don't have much of a commute either, do you?
I don't. I just have to walk across the street.
See, I like taking the train for about an hour. I actually read TWiV papers. I do show notes. I can get work done. So I by the way, I don't listen to podcasts anymore because I don't drive. And I figure, why listen to a podcast on the train? I could do work on a laptop. So an hour, and then I walk 2 blocks. I'm in heaven, except when it floods. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 83 degrees, and I would call it, I guess, high overcast. Occasional something kind of like sun peeks through. It's a good day to be doing this.
Does it rain a lot in Austin, or now and then?
Now and then.
Is there a rainy season?
Yeah. I haven't quite figured out when it is. I think it's like March-ish. There's been a lot of rain this year. I think the average rainfall is about 35 inches. We've had a lot of excess rainfall this year. It's supposed to thunderstorms supposed to roll through tonight. We'll see.
So New York City, average rainfall is 47 inches. It rains 119 days a year. That's a lot of days. Wow.
In Gainesville, it was 52, 53 inches. Rained in the summer. Rained every afternoon.
Yep. Florida's like that. And then the sun comes out.
Yep.
All right. We have a episode that's been in the planning for a while today. We are going to talk about pox viruses, specifically smallpox and relatives. But Rich has been talking about this for a while. We actually have a second episode, which will come later. But I said, let's do this in our effort to wean everyone off of COVID.
Diversify.
We want to diversify. And people have responded well. They're enjoying the other episodes. So today we're going to do a little pox virus with Rich Condit, who of course spent his career working on Vaccinia virus. And well, his friends, many of his friends worked on Vaccinia Virus as well. They used to play golf together.
Yep, the Pox Virus Invitational Golf Tournament. I was thinking about that the other day. That hadn't happened for a long time since people retired, you know.
Yeah, sure. But you would do it at a meeting. I know that we'd be
Yeah,
at breakfast
meeting,
one morning, and then everyone's at breakfast ready to go to the first session, and Rich and McFadden and a few others have their bags over their shoulder. I'll see you at the first session. Yeah.
when I was the program chair at ASV, the rule was that you could not have the Pox Virus workshop in the afternoon. It had to be an evening workshop. Okay, and we tried to indoctrinate the downstream organizers to make sure that happened, but they didn't get the message.
I never got the message. You could have told me directly.
I suppose. Yeah. You were further downstream. The immediate downstream people didn't. Kind of tried for a little while. Didn't happen.
But I think it speaks well of your field that your friends are pox virologists.
Pox virology was remarkable in this regard.
Yeah.
People were very generous, very forthcoming. People helped each other out. Even there were a couple of competitions at one point that were a little bit unpleasant, but there was also a lot of competition that was really quite vigorous and friendly at the same time. But
that's awesome.
But I don't think anybody ever denied me information or reagents or anything like that. Everybody was always happy to help everybody else. We're all in the game together. It was great.
Yeah, I remember.
There are other fields I can think of where the invitational golf tournament would have paramedics showing up.
Or if it happened at all. I remember one year Rich was at a coronavirus session, and he said to me afterwards, I can't see you guys playing golf together.
Yeah, I don't think the herpes virus invitational would have been a thing either.
No. That would have been a different sort of gathering.
All right, Rich, it's all yours.
Okay. Well, you guys are going to have to help me out a lot with this. At any rate, okay, so cut to the chase in a way with some introductory comments. I picked out 4 articles, one of which is a research article from Science. I'll even tell you what it is. It's called Diverse Variola Smallpox Strains Were Widespread in Northern Europe in the Viking Age. And I'll come back to that. And then there's 2 other articles that are basically comments on this article that were written either in the same journal or a different journal that try and provide some perspective on this. One by Laura Spinney, a science journalist, and another by a friend, Antonio Alchemy, who is a pox virologist. And we'll reference those as we go along, in and out. And the last is an interesting thing. It's a 1988 article on basically the history of smallpox, a brief history of smallpox, by Don Hopkins, who currently is 80 years old and was a prominent figure in the smallpox eradication campaign, and so has boots on the ground experience with smallpox eradication and the whole thing. And I'm using that to sort of fill in some of the history.
The history is also an excerpt from his book, Princes and Peasants, Smallpox in History.
Yes. So let me introduce, I'll try and introduce Kathy style, the paper, by saying its goals and importance and what it does. The goal is to try and get a grip on when smallpox arose, and I suppose a little bit on where, and a little bit on how it evolved from that.
This is a question that has had a lot of competing mutually exclusive theories floating around.
Yeah, and we will detail that as we get into it. The approach is to isolate DNA from humans dating back centuries and sequence that DNA and look for smallpox sequences. And the answer, in very brief, we'll go over all of this in detail, is that they can find variola virus, that is smallpox virus, the virus that caused smallpox, dating back to the Viking Age, actually isolated from Vikings in 600 CE, okay, which puts a new place mark on the evolution of these viruses, as I will emphasize. Why is this important? Well, I think it's of pure academic interest to start with. Second, and I think this is, it's relevant to our whole SARS-CoV thing. We need to know how viruses get into humans, all right, and how they evolve once they're there. And smallpox is one of the classic human pandemic epidemics, okay, or if not the classic. And so it's a perfectly appropriate one to be looking at.
And also, because this paper is sequencing based, they can draw conclusions about another critical question, which is what happens after the virus gets into people? How does it change?
Right. So I want to give a little bit of background on the virus and the disease. And, I think it's probably, I'll bet you it's TWiV 26. The first time I was on, Vincent had me on to discuss pox viruses. It's back there somewhere. Okay. So, and that pretty much is for me for an hour talking about pox viruses. So you could go back to that in background, but I want to do, and we've done this briefly, periodically, but it's appropriate to at least summarize the whole thing now. So, smallpox virus is the virus that causes smallpox is called variola. The disease is smallpox. Okay, the virus that causes it is, but I'll say smallpox virus a lot. It is one of a family of viruses called pox viruses. They are double-stranded DNA containing viruses that have the somewhat unusual property of replicating in the cytoplasm of infected cells exclusively. DNA ought to do its thing in the nucleus theoretically, but this guy's decided to skip the bureaucracy and set up shop in the cytoplasm. It's one of what we've talked about before, nucleocytoplasmic large DNA viruses. It's, historically the original one, but others that we've talked about include the giant viruses and some others. The pox viruses themselves have an extremely broad host range that includes basically any vertebrate you can name, and invertebrates as well. They're large. They are about 200 on the average, 200 kilobases of DNA. I think it ranges throughout the whole family from anywhere from 100 to 350. And if you figure that 1 kilobase of DNA is enough to make a one gene, we're talking on the order of 200 proteins. So smallpox disease, there are literature records. There are descriptions of things that seem like they must have been smallpox that go back to a couple of thousand BC. What are we calling as BCE is what we call it now. Is that right? BCE.
Yes, before Common Era.
Before the Common Era. Okay, thank you. But from descriptions, it's always a little difficult to sort of pin it down. You don't. What you'd really like is hard physical evidence, which this paper is going to talk about. But certainly, the disease was endemic. It was widespread globally, essentially, certainly by the 15th, 16th, 17th century. Okay. To the point where, in particular, if you lived in an urban area, you could be pretty much guaranteed that you and your children in particular were going to get smallpox at some time during your lifetime. It was a horrific disease, characterized in its sort of peak stages by these blistering lesions all over your body and, in fact, in your mouth, an oral cavity, and et cetera. Extremely painful, high fever, malaise, et cetera. Reasonably contagious, but it was around enough so that so that everybody was going to get it at some point. Transmitted primarily as a respiratory infection, but it was transmitted also on fomites, so that clothing and bedding, you could, if you came in contact with it, you could pick it up. People got a respiratory infection. They got a primary viremia. Then they got these blistering lesions, and the case fatality rate was about 25%. So if you had 4 children, all of them were going to get smallpox. One of them was going to die on average. If you survived, you were left with these scars all over your body. And one of the common side effects also was blindness. There were techniques for trying to control this from early on, and I'll only highlight the ones that were ultimately successful. The first of which went by the name inoculation, which meant, and I'm looking here, which meant taking material from a person who had an active case of smallpox and scratching it into the skin of someone who you were trying to immunize.
And this material was usually scraped from the lesions, right?
Right. Yes. Scraped from the lesions and scratched into the skin. And basically, you got a pretty horrific reaction on your arm. But my understanding is that it was significantly less likely to establish a viremia, that is, spread through your blood and spread through your body. So the lesion on your arm would heal up, and that would confer immunity to smallpox. This dates back to, it says that inoculation was apparently, this is from the Hopkins article, used in India since before the birth of Christ. So that actually, if that's the case, and this is from literature records, but if that's the case, that says smallpox was around BCE. Okay.
Right.
It was inoculation thought to have been introduced in China in the 11th century. Importantly, the best documented was that it was introduced into medical practice in England from Turkey in 1721. And widely, that practice spread through Europe and the United States, in fact, for the next 70, 80 years.
Yes, you can actually, if you look at a lot of immunology textbooks, they often have a picture of Lady Mary Wortley Montagu, who was the ambassador's wife, who is often thought of as the person who did that introduction. And I know that there also are citations of Onesimus, who was an enslaved person with Cotton Mather, who brought it to Cotton Mather.
So the downside of inoculation was that it had about a 1% case fatality rate. Okay, so talk about side effects.
Which it beats 25%, but it
beats 25%. So it made it very well worthwhile, but it was still not as good as you would like. So that brings about vaccination. And the short story is that Edward Jenner in 1796, he made he was a physician. And in fact, he was part of his practice was doing inoculations. Okay. So he had all of these concepts in his head that by transferring this stuff that of course, they didn't know what it was, but this pus from a lesion. You could create a similar lesion in a naive individual and confer immunity. And he also made the observation, and others had made this observation as well, that there was a disease prevalent around called cowpox, which was a similar thing where people got, milkmaids in particular, got lesions on their hands from milking cows. There were similar lesions on the cows. And the observation was milkmaids seldom got smallpox and were refractory to inoculation. Okay. And so in his publication, Jenner documents this. And in fact, we had an episode later on TWiV where we went through, Dixon and I went through that publication in detail, Jenner's inquiry.
Yes.
And so Jenner did the experiment of inoculating a kid, 12-year-old James Phipps in 1796. And then 6 weeks later, because the practice of inoculation was commonplace, he actually challenged him with smallpox and tried to inoculate him, and the inoculation didn't take, so he knew he'd gotten it. And so the practice grew. That practice spread and grew because it was greatly desirable to inoculation, to the point where it was in common practice globally. The virus that is used to do that is something that came from nature. Okay? We call it cowpox because originally cows were involved. Okay? But if you read Jenner's inquiry closely, there's horses in there too. Okay? And it's also clear nowadays that what we call from sequence, that what we call Vaccinia virus, the virus that was used to vaccinate people, vaca, by the way, is the Latin for cow. That's where that comes from. That Vaccinia virus is different than cowpox. Okay. And the next episode, somewhere down the road here, we're going to have another episode about the evolution of the vaccine virus, Vaccinia. Okay. But that's not ready yet.
Well,
So-
on a past episode, we talked about the link of it to a now apparently extinct horse virus, horsepox,
Right.
and that's just a fascinating story that we may have accidentally eradicated horsepox, but we got Vaccinia out of it before that.
And that whole story, the upcoming episode, that whole story is evolving. Okay.
Yeah.
With people sequencing stuff from anywhere they can get it. Okay. To get information on the evolution of this virus.
Yeah, I was going to ask you a question, Rich, and I'm realizing it may be more relevant for this other episode that you're mentioning about some stories about sort of questioning some of the exact details of the original Jenner story. There was the sort of history of science story by Boylston in 2018, and another one that I often cite from NPR around that time. Should I hold that thought until later, or is that?
What's the thought that we're going to hold?
So the idea that maybe Jenner had made some observations from others who were doing things using materials from horses.
I don't think you need to hold. I don't think you need to hold that thought. Among other things, Jenner was not the first.
Yeah.
Okay, he was the first to publish.
Right.
Okay, which is a common historical lesson. But no, I would actually yes, maybe hold that thought from the point of view that when this other episode comes up, if you've got links to those episodes,
I just pulled up both the links. I can put them in the show notes.
Sure. Yeah,
but-
that'd be great because I haven't
Okay.
heard those. That would be very interesting.
Also, I remember that we teach something about the milkmaids having the peaches and cream complexion because they didn't get the pox. And so I was looking for that and found a story that Goats and Soda from NPR.
That might be the same one.
that the milkmaid thing is a myth.
Yep,
yeah.
that's the same one.
So
Yeah, because I can remember looking for that about complexion like a milkmaid and coming up empty-handed in the past.
Mm.
But the whole thing about milkmaids being refractory to inoculation is not a myth, okay? Because there's a significant fraction of Jenner's inquiry that looks into this and characterizes it.
And it may well be that milkmaids were free of smallpox, but given the time period and the health conditions and the fact that milkmaid was not exactly I'm imagining highly paid noble calling, they may have had other skin issues that precluded grouping them as being of fine complexion.
So at any rate, moving forward, we get into the 20th century, and a lot of countries had individually mounted campaigns to, I want to make a distinction here. We use the word elimination when a disease is removed or gotten rid of in a country. Eradication is when you get rid of it globally, okay, and actually get rid of the virus globally. And even in the 1950s, there were still over 90 countries worldwide where smallpox was endemic. And it was around 1966 that the World Health Organization established a budget to implement the smallpox eradication program. And at that point, there were still 44 countries reporting smallpox. So that was 1967. The last case of smallpox was reported, the last naturally occurring case of smallpox was reported in 1975. And the disease was declared eradicated in 1980, which is probably one of the most magnificent accomplishments that humans have ever done. It's important to realize that there were special characteristics of smallpox that made it low-hanging fruit for eradication. It was easily diagnosed. I'm taking this now, a lot of this from the Hopkins article. It was easily diagnosed, and there were no asymptomatic infections. You could tell whether a person had ever been infected or ever been vaccinated because they had a scar. And no animal reservoir. The duration of infectiousness was short. There was no asymptomatic transmission. Natural immunity was complete and lifelong, and there was an excellent vaccine. So,
and that excellent vaccine was not directly related to the causative agent. So you could continue making the vaccine, and you had no issues with the vaccine containment or anything like that because it was a different virus.
Right.
Unlike polio, which
is the same virus.
You have to get rid of it all once it's eradicated. Rich, can I make a few comments?
Sure.
So this history article, I think, was really good. This history of smallpox, 10 years gone by Donald Hopkins. It's really, really good. I want to point out just two parts near the end where he talks about eradication. He says that, and this is particularly useful in light of the narrative around COVID-19. He says, despite earlier skepticism of many otherwise knowledgeable critics, smallpox was eradicated. I can imagine people saying it wasn't, just like people say things today. Eradication is a good thing for public health, but it should be chosen wisely. And he notes 4 diseases that are appropriate targets for eradication: guinea worm, polio, yaws, and measles. And he writes that the fact that kids are still crippled by yaws, which can be cured by a single injection of penicillin, is a global disgrace. These are words that people don't use much anymore for some reason. But
because we kept lowering or raising the level of disgrace we were willing to tolerate is what happened.
I guess so. But I love that kind of thing, that's what it is. And then today it doesn't make an impact on many people, but it is a disgrace. And then he says, to eradicate, you need international cooperation, which he says even back then in the 80s when he wrote this, or 10 years after smallpox eradication, 90, I guess. He says, we only have to look at the squabbles between the U.S. and the French over discovery of HIV, the disinformation the Soviets put forth saying HIV was made in a U.S. lab, and other things to demonstrate how far the world is from acting on this lesson of cooperation. And we're still not there. We still don't cooperate, right? And then finally, he talks about the remaining stocks in Atlanta and Moscow. And I wanted to ask you about that, Rich. But first, he ends by saying, as we eradicate smallpox, we have a new virus coming on the scene. So we shouldn't be smug about eradicating anything because we're going to have new ones. And it says future historians will remark on this irony, and they will likely be amazed and saddened to see how closely the responses of some of our contemporaries to the new virus match the fear, ignorance, and xenophobia and political opportunism formerly engendered by smallpox. COVID is nothing new, folks.
Nothing new. Absolutely.
This is an article from 1980 predicting that. Yeah.
Yeah, and there's that famous engraving from Jenner's time.
Yes.
The side effects of vaccination, including people growing horns and cows out of their butts and stuff like that.
So, Rich, is it correct that the known stocks of smallpox virus are in Atlanta and Moscow? Is that correct?
Correct.
Do we think there might be other unknown collections?
I mean, we can think that if we want. Okay. At the time, when smallpox was declared eradicated within a very short period of time, a couple of things happened. First of all, and this is interesting, vaccination was discontinued. The recommendation became not to vaccinate people because the risks of dying from the vaccination were greater than the risks of dying from the disease. Isn't that remarkable?
Yeah.
And at the same time, a global effort was made, the same as it is with polio, only I think a lot less complicated than with polio, to tell anybody who is working with smallpox, destroy your strains.
Yeah.
Or if you have any that are useful, give them to us. Okay.
But this was happening at a time when there were a lot fewer virology labs and it was simpler.
And I think also, well, I don't know, I was going to say a lot fewer people working on smallpox. On the other hand, the pox viruses at the time were one of the sort of marquee viruses to work on because of the disease issues. Not only that, but you were vaccinated and there was none of this biosecurity stuff. You worked with your smallpox virus, mouth pipetting the stuff out on the bench. Okay, so
and I remember Grant McFadden telling us that it's not like there are a couple of vials in each place. He said it's a whole collection of different isolates.
Right.
So it's quite a useful collection. Just in turn, and they should at least be sequenced, which I think hasn't been done yet. But he said many vials. So.
It's a repository of hundreds of different strains or hundreds of different viruses collected in a documented fashion from different places. So there's the potential there to learn things about virulence and virus evolution and et cetera. Now, actually, with respect to the molecular biology, I suppose I'll get to this, but I'll introduce it now. I do want to say that in something like smallpox, for example, there's basically two fundamentally different kinds of genes, if you like. There's in the middle of the genome, it's a linear double-stranded DNA molecule. In the middle, roughly half of the genome, there is a cluster of on the order of 100 genes that are very highly conserved in all of the pox viruses. And these are all the nuts and bolts: DNA polymerase, RNA polymerase, structural proteins, etc. On the flanks, on either end, another roughly half a genome's worth, a quarter on each end, are genes that are highly variable as you compare one virus to another. And these are largely genes that are involved in host range and pathogenicity. More likely than not, involved in functions that tweak the cellular and the organismal response to the virus infection. Immunomodulatory genes of one sort or another, either targeting the innate or the adaptive immune response. And these become important in this paper. So let's see. I want to actually just read the first paragraph out of this research article summary because it really sets the stage nicely. We've covered some of this, but this really summarizes it. Variola virus, the causative agent of smallpox, is estimated to have killed between 300 million and 500 million people in the 20th century and was responsible for widespread mortality and suffering for at least several preceding centuries. Humans are the only known host for variola virus, and smallpox was declared eradicated in 1980. The timeline of emergence of smallpox in humans is unclear. Based on sequence data up to 360 years old. That is sequences from
Samples 360 years
from,
old,
that
yes.
were 360
They were
years
not
old.
sequenced 360
That's right.
years ago.
That was an awkward phraseology there.
Yes.
The most recent common ancestor of variola virus has been dated to the 16th or 17th century. That's a paper within the last few years, and it really kind of struck a nerve in people because they said, well, no, wait a minute. All the records say it was around for much longer than that, okay? This contrasts with written records of possible smallpox infections dating back at least 3,000 years, and mummified remains suggested of smallpox dating 3,570 years ago, okay? So their objective is to use ancient DNA sequencing to try and pinpoint better when smallpox entered the human population.
Right. So there's this famous Ramses V, who was a pharaoh of Egypt, and I'm just looking at the history paper, which the Hopkins history paper is open access. And Ramses died in 1157 BC, and looking at the skin of the mummy, it has features that you could say look like smallpox.
Notice Alan is sort of waffling there because I've stared at this picture a lot, and he's not looking real chipper.
No.
So
it is a mummy, but the skin is not exactly perfectly preserved. I mean, this is from a few thousand years ago. So,
but didn't we do a paper where they thought a mummy had smallpox, but then when they sequenced it, it turned out to be hepatitis B virus, right?
I seem to recall something like that. Yes. By the way, before I get any further into this, let me say that this paper has two co-first authors, Barbara Muehlman and Lasse Vinner, and it has three corresponding authors, Eska Villerslev, Terry Jones, and Martin Sikora, and they're from all over the place. University of Cambridge, University of Berlin, German Center for Infectious Research, University of Copenhagen. A lot of these are from museums here and there because they're getting samples from, ancient remains. And I won't go through all of those, all the places, but those are the highlights. England, Denmark, Germany, mostly. Okay, so.
And they're going to sequence a lot of samples here. They 1,867 humans, and then they've got samples from all these different museums and such.
Right. So we screened high throughput shotgun sequencing data from skeletal and dental remains. We've been through that. These are good places to get DNA out of remains of, as Alan said, 1867 humans living in Eurasia and the Americas between 31,630 and 150 years ago. Okay, so that spans a long period of time.
So I have a quick question. Do we think that this method would sort of specifically be useful for looking at pox viruses? Or would you be able to look at some other types of viruses with this same kind of method?
You ought to be able to look at just about anything, any DNA virus.
Any DNA virus.
Okay. RNA viruses, not so much. Okay.
Would you have a better chance with pox because it's big? I mean, I'm thinking maybe a smaller virus, the genome would be more damaged. But
or would the virion be kind of a little bit more stable? I mean, people think about it as being kind of stable.
I can't really answer that. I kind of doubt it. I think the critical element here is DNA and having the right primers, okay, and targeting in on it. But I can't answer that directly.
Okay.
They say they identified a total of 26 samples with reads hits to variola virus. Of those, 13 they enriched the DNA with a hybridization approach where you hybridize the DNA from the sample to pox viral DNA and then pull that out of solution to enrich for the sequences. And then here's sort of the punchline. 11 of the 13 individuals are dated to 603 to 1050 CE overlapping the Viking Age, which they say is 793 to 1066 CE, and are from Northern Europe, Western Russia, the United Kingdom, and 2 are from the 19th century from Western Russia. So for those of you who are not into the Viking Age, we're talking about Scandinavians, primarily from Denmark, I believe, okay, doing their best to expand, okay? And Vikings specifically is a term to describe the Danish who actually sailed out to on conquest, missions of conquest of one sort or another. That includes Northern Europe and the UK and Russia and etc. And they left behind corpses.
Iceland, Greenland, Vineland.
Yes.
Yeah.
They left behind corpses, some of their own, a lot of others as well.
A lot of, yes.
So 4 of these 13 individuals, they were able to get near complete sequences, one of which they got 99.98% of a complete variola sequence. Okay. So right away, we have at least one of these that comes from about 600 CE. So right away, the old idea, or the more recent idea, that variola may have showed up in the 16th or 17th century is disproven, okay? And we have variola on the scene by at least 600 CE. That doesn't help us right away with the notion that it may have been around thousands of years earlier than that based on historical records.
Now, an important thing to remember about these, so people have obviously noticed the funnel that went on here. You had more than 1,800 people initially who you funneled down to this basically dozen samples, and now we've got 4 full sequences. We're dealing with a very, very small subset of the total. But what I wanted to point out is that these would only be people who had active viremia at the time of death, I think, if that's correct, right?
That's what the authors state, which I thought was, I hadn't been thinking about it when they said that. Makes perfect sense because this is an acute infection. Okay, you get it and it goes away, and there shouldn't be after you've recovered from the recovered from the infection, there shouldn't be any latent material hanging around. And so the authors even go so far as to speculate that the individuals from which they're recovering variola DNA actually died from the infection.
Died of it, yeah.
Certainly, they were infected when they died.
They either died of it or they died with it. Yes.
Yes.
So I wanted to ask a little more about that because, at first I was thinking, well, any card-carrying DNA virus is going to cause persistent infections, but the pox viruses having a different replication scheme being in the cytoplasm, maybe they don't have that opportunity. But that same thing that you were just describing, where the authors talk about making the assumption that the virus did not lead to a persistent infection. So the fact that they found it in the teeth and in these very hard bone structures near the auditory system indicates that they had some acute infection at the time.
Actually, this raises a really interesting point because certainly I think of smallpox as being an acute infection, and I know of no evidence to suggest that it was anything other than that. However, there are other branches of the pox family where and I'm thinking in particular of some parapox viruses that infect aquatic mammals, seals. There was a project in my lab on this for a while, where there are recrudescent infections. And they show up, among other things, because seals that are, we got involved in this because there was a seal rescue outfit in the Marin Headlands, north of San Francisco, that were really upset because they would bring in these seals that were rescued and they would turn up with seal pox. And they were concerned that the seals were being infected in the facility because they would come in looking reasonably clean. And one of the conclusions that we came to was, no, it seems like there can be with this virus recrudescent infections. Now, I don't know exactly the mechanism of that, but it implies that there could be some sort of persistence. So I wouldn't rule out the notion that, and some of my poxy friends may write in about this, I wouldn't rule out the notion that there could be, at least with some members of the whole pox virus family, viruses that caused some level of persistence. But as far as smallpox is concerned, I'm not aware of any evidence. I would think it would be acute. I just wanted to elaborate for a minute here. I remember we've done a couple of these papers with looking at human remains, and the archaeology is as interesting as anything else. And this paper has a picture of one of the skeletons, and Laura Spinney's article elaborates on one of the sites. Let me find it here. Oh yes, one of the skeletons was unearthed from a mass grave in Oxford, UK, and is thought to have died in the St. Brice's Day massacre of 1002, when the English king Ethelred the Unready ordered the extermination of people identified as Danes. Okay, so yeah, there's Kathy's got the picture.
Ethelred the Unready, when we knew how to name our politicians.
Supposedly, there's a dagger that we can see, and I'm not sure if it's.
I was not able to identify the dagger either.
Yeah. Is that a dagger which you see before us?
But it's the full page in the article.
Wow.
Okay, so now we've got sequence from a number of individuals and complete sequence from a few. And we can do a couple of things. They do a couple of things with that. First of all, of course, is to draw a phylogeny. And I won't go into this in detail, but the most interesting thing that comes out of that is that for the four ancient sequences that, and I think these are pretty reasonably spread out in time and space. As a matter of fact, you can see that in the diagram. The four ancient sequences represent a variola virus that, from genetically, has not previously been identified. And we said we've got hundreds of these things, okay, in the modern collections in Moscow and in Atlanta. These are four strains that have not been previously identified and, based on their phylogeny, presumably disappeared. Okay, so the idea is that there have been, in time, strains of variola virus that were running around in humans that eventually, for one reason or another, died out. And that's contrasted with the modern, what they call the modern version of smallpox, and that their sort of identifying sample for that is this one that we spoke of earlier that was taken from a mummy from the 16th or 17th century. So it can be dated as like 350 years old, and the phylogeny on that suggests that the most recent common ancestor of the modern pox viruses or variola viruses dates back a mere 450 years, which really surprises me. And yet the most recent common ancestor of all of these things grouped together dates back about 1,700 years. So now we're getting back to 1,700 years of 400 CE. But they have another figure where they graph all of this on a graph that is where they're looking at a disruption of genes that I'll get to, and years into the past, where they extrapolate. That gives a different sort of perspective on the evolution of these things. And if you extrapolate that back, they don't make a big deal out of this because I think it's pretty shaky. The intercept on the x-axis, where there are no genes disrupted, and that's another way of thinking about potentially the primordial variola virus, is 4,000 years ago. So now we're getting back into close to the Egyptians in some of those records. Okay, so I think there is some support at least, for the notion that these viruses have been around for thousands of years. But there's another little bit in the Spinney article that I really enjoyed, where she says that an author, a scientist named Willerslev, who's done a lot of this ancient DNA stuff, thinks his group has screened enough individuals from the Bronze, Neolithic, and Mesolithic age, stretching from around 15,000 to 1200 BC, without finding variola, to say that it's unlikely that smallpox was circulating widely before 3,000 to 4,000 years ago. So we're closing in on this.
Yes.
Okay. It was there in 600 CE. It was probably there before 600 CE. It was probably not widespread, at least, before. So 2000 BCE would be 4,000 years ago. So we're sort of closing in on this. Interesting. Okay. So one other thing that comes out of this paper relates to these immunomodulatory genes. And they go into a long wrap on this that frankly loses me at places. So I will summarize as best I can. You can surmise things about the relatedness and evolution of these viruses based on which of these immunomodulatory genes is present in an active form or absent altogether or inactivated. And a fundamental concept here is that the primordial pox virus, the idea is that the primordial pox, and I don't know if this is true or not, but it's a nice way to think about it. The primordial pox virus, all of these accessory genes were probably active. And there are good examples of this. The camel pox, no, it's not camel pox.
I think it's gerbil, right?
Gerbil pox,
Yeah.
one of them, has almost all of these things active, okay? Not only that, but there seems to be at least a vague correlation between how many active accessory genes you have and your host range, which makes sense to me. Okay, it's truthy.
Yes.
Put it that way.
It has that truthiness to it.
It makes sense that if you had a bunch of accessory genes that can tinker with all sorts of different cellular pathways, you might have a better chance of infecting a wide variety of hosts.
You'd be ready for whatever kind of.
And as you lose those immunomodulatory genes, you become more specialized. Okay.
You would not be like the unready king. Yeah.
Right. And I think that's basically the take home message of this analysis of these accessory genes. Beyond that, they make a clearer distinction between ancient and modern pox viruses, because they can identify essentially banks of genes that are all either present or inactivated in all of the ancient guys, but not the modern ones, or the other ones, present in the modern ones and not the ancient ones, or absent in both, or present in all of them. But it gives another distinction between the ancient and modern pox viruses. And now, that's where go ahead.
In terms of these genes getting lost, I mean, I can think of a couple of different reasons why that might happen. It might be that the virus was spreading fine and getting around beautifully in humans and didn't really need to be adapted to other hosts. And so the genes just gradually accumulated mutations because they weren't necessary. It's also possible that they were counterproductive. And this is something that I think the Alchemy article riffs on a bit, that these were making the disease so much worse in humans that it was spreading poorly because it was killing off its hosts. And so it lost those, which might have been virulence factors because they were over suppressing the immune system, which is another possibility, but those are just theories.
And I would say that once again, in the realm of theory, and I'm not going to detail this at all, in this article, there's discussion of the mechanism of action of several of these genes. Okay, but it's known in several cases what they do, and there's speculation as to how the presence or absence of such a gene might change the property of the virus. But I want to say that as far as I'm concerned, and I may offend somebody here, but as far as I'm concerned, with all of this information and all of these genes and all of this stuff, we don't know what controls virulence or host range in the virus, because it's extraordinarily complex. I think both host range and virulence are actually, at least likely, and I think actually, based on what's been published, multigenic phenomena. So you can't identify this or that gene with a few exceptions, this or that gene that controls host range as you see it occurring in nature, or in immunomodulation. And so we're just going to have to work on it.
They make this argument that these viruses have become more virulent in humans because they have lost some of these immunomodulators. And they say this seems counterintuitive. Yes, it's quite counterintuitive. But they make then the argument that if you lose the immunomodulator, you get more virulent because the host is actually what's responsible for the severity of the disease, right? So it's kind of a weird argument, because then the virus is not becoming more virulent. It's just the host immune response. But it's based on really one of these orthologs. I think it's an ILB1 receptor ortholog, which if you inactivate it in mice, if you activate that gene in a virus and then infect mice, they get a febrile disease. But if the soluble receptor ortholog is present, they don't have a febrile disease. So from that observation, they make this large conclusion that
big leap. Yeah.
It got more virulent in people. And I don't think you can even say that, right? It's just complete speculation. I mean, if you say in gerbils, I think in gerbils, the virus is not virulent, but in people it is. That doesn't necessarily mean that it got more virulent in people. Anyway, it's a lot of hand-waving, I think.
Yes.
And it's a good part of the paper that they spend talking about this as well.
Yeah. Right? And I think, you're faced with all these accessory genes that you have a lot of information on. Okay. So what are you going to do? You can't do much more than speculate. And there's a lot of fodder for speculation, but I think it's speculation.
And they include, so there's this bit towards the end where they're talking about the historical perspective, and they have kind of a counter argument to that discussion. I mean, it's like there's a debate within the authors even that there's a lot of uncertainty because of the initial sample size and then the detection rate. And they basically say you can't really use our detection rate as an estimate of prevalence of smallpox in the ancient world, and we can't really draw any firm conclusions about the virulence of this virus because we don't have real-time samples taken during an epidemic that was occurring then.
But it certainly would be interesting, right, to take Vaccinia virus. Does Vaccinia infect mice, Rich?
Yeah.
So
one can infect mice with Vaccinia, yes.
Or just some pox virus that causes a good infection in a rodent, a mouse, and then systematically remove the modulators and see the effect on, and then at least you can get some indication of whether there's any sense to this idea or not, right? I'm not sure anybody would do that.
And I don't know if that's ever been done, like, as you say, systematically. Okay? Take them out one at a time or in groups or whatever,
exactly.
and see what happens to the virulence. Certainly, there's a huge body of literature on individual genes.
Sure.
What they do and what happens if you have them or don't have them. But of course, it's really complicated because I don't know that if you want to do it systematically, you got to start off with something that has as many genes intact as you can, and one by one, and then in groups, knock them out. That would
maybe the
make sense.
gerbil virus would be a good place to start. Something like that.
It's an interesting idea, but I'm afraid now that this has gotten a lot of publicity. It's from 2020, but I'm afraid people are going to conclude that without reading it in much depth and not knowing the field, that, smallpox got more virulent, therefore COVID should get more virulent too, right? And I don't think that's correct, but...
Well, and then, go ahead, Brianne.
I was just going to say, I feel like in some ways there's sort of this sort of Goldilocks argument on the virulence that, there are some ways that it got more virulent and some ways that it got less virulent. And none of them are, a lot of them are all speculation.
I don't know which paper it was in, but they say the only real evidence we have is from the myxoma experiment in Australia, where it lost virulence as the virus went through the rabbits in that first year. And I think that's fair, right? Because that was an actual experiment that was done.
Yeah.
Right.
Yeah, and I think that might be in the Alchemy commentary, where there's this statement early on: evolutionarily successful viruses replicate and transmit without causing host pathology.
Yeah,
which I put a note next to in the paper. Yellow fever virus would like a word with you. I can give you a list of a dozen that cause pathology and are evolutionarily successful. So there's this long-standing notion that I thought we had kind of put to bed years ago, that viruses always evolve with their host for a lesser pathogenesis, but that's not consistent in any way. I mean, some viruses have gotten to the point where they're virtually symbionts, but there is an equal and opposite body of the virome that's highly pathogenic, and it all depends on the mechanism of spread and what that virus needs to do to get to the next host to continue propagating its genome. And if it's a vector-borne virus, then incapacitating the host and killing them within days is not a problem, because that means the host is not able to swat away the mosquitoes that are going to carry it to the next host. If it's a respiratory virus, then it probably needs the host to stay ambulatory while it's spreading. So it's a question. It's a complex situation, and as Rich has pointed out, we don't have the data to draw firm conclusions about what smallpox has or has not done over time, because we just don't have the past.
Well, this article, though, concludes, he says, a question remains unanswered. Why did variola evolve towards higher virulence in humans?
That hasn't been proven.
No, not at all.
Not supported at all.
Not at
This
all.
Is from the Alchemy commentary. Yeah.
It's real alchemy.
Right.
It's real alchemy. Yes.
Right. So I'm not. I had all the same things highlighted in my version of that. So these stuck out at us. And by the way, that's same article, he does say virulent pox viruses are believed to have evolved from a common ancestor encoding the full collection of about 200 genes, likely similar to cowpox virus, which causes limited pathology and infects a broad host range of species. Now, whether that includes gerbil pox or not, I don't know. Some of my, as I said, poxy friends may ping me on this one. But I should point out, as long as we're talking about it, the cowpox virus is not a virus of cows. It's a virus of rodents, okay, that spills over into cows now and then, okay? So gerbil pox could be a close cousin.
The figure in this paper is very nice, the alchemy.
Yes.
It's nice.
Yes. Because he contrasts what has been the dogma about the evolution of smallpox virus originating in Africa and spreading to the Middle East and Europe, with what has been observed in this paper, and speculating that, in fact, even the geography of the origins may be different, or at least this notion that it was around 600 CE in Northern Europe and Scandinavia and Russia has to be taken into account.
Yeah.
I have one complaint about the figure, and that is that they could have used brighter colors to distinguish their lines. And
I had to
dots.
dig around. Yeah.
I mean, I had to enlarge it and
Dark green and purple and light green and blue and
black.
So Rich, the paper that we've been talking about kind of comes to this idea about origins being about 4,000 years ago. So we have this idea of time, but I think that one thing that someone who has not read a lot in all of this might least ask questions about is sort of any other things about the spillover, like spillover from where, what animal should I stay away from if I to stop other spillovers?
I don't know that we have a lot of information from that about that. We've mentioned a couple of viruses. These are pretty common in rodents, and there's a lot of spillover from rodents into other species. So that is a reservoir that certainly, if I were trying to answer that question, I would be looking at. Okay, but I can't speak to it much more than that. And I do want to reemphasize that the solid number is 600 CE,
Yes.
and it was
Right,
prevalent enough so that they talk about the sample. It was roughly 2% of the samples were positive. Okay, so that's a lot of people. Given that's a pretty high percentage in my mind, given that this is an acute infection with a likely lethal outcome. So I think that speaks to how widespread it might have been. And so I think you can certainly go back to that era. But I think the combination of looking at the loss of the accessory genes and the historical record and the other archaeology that says that it probably wasn't around before 4000 gives you a window of, between 2000 BCE and maybe 500 CE, where we're talking about, I don't know whether that's when the spillover happened, assuming it was a spillover, must have been, okay? And some period of time for it to really become established.
Can you fit in the Indian inoculation data that I had the feeling was several thousand years BCE?
That was no, that was let me look that up again. That was around 0, I think. Where's that? That's in it's in Hopkins' paper, and I will have it here. It's it's in the inoculation section. I'll get here, inoculation. Inoculation apparently used in India since before the birth of Christ.
Okay. So it could still fit within that 4,000.
That fits within the window.
Right,
Okay. And it's not very specific there. I ought to probably grab down the big red book. One of the charges to the people who did the smallpox eradication was that when they were finished, they needed to write it up. And so there is this tome sitting on my shelf that's a couple of thousand pages long.
Wow.
Called Smallpox and Its Eradication by, if my new bionic eyes, I can almost read all of the authors. But the first two are appropriately Frank Fenner and D.A. Henderson. D.A. Henderson ran the whole show. Frank Fenner wrote the book. Okay. He was involved in the show too, but he wrote the book. And that'll have a lot of this history in it as well.
So I'm going to put a link to an interview I did with D.A. Henderson a while ago. He talked mostly about smallpox eradication. So he came to ASM headquarters and I chatted with him. I want to show this slide from my lecture because it kind of jives with this papers. And this is about the origins of smallpox virus. I think I can pin myself to make this bigger. Hang on here. Let's try that. Pin. Do you know? I have to spotlight it for you guys, right? Hang on.
I see it.
You see it big? Or?
Yes, sir.
Big, right?
It's in lieu of your video.
It's in lieu of you, yeah.
Okay. But so this, I don't know what this is. This is from a paper. It says phylogenetic analysis of related viruses suggests emergence of smallpox 3,000 to 4,000 years ago in East Africa, transmitted from camels, which is not in these papers. And camels came into Africa from their origin in the Americas. They came over the land bridge. They came into the Arabian Peninsula. And apparently they got infected with a ancestor of smallpox from gerbils, which we see in these papers here. The gerbil range is shown in kind of Central Africa there. So the idea being gerbils, camels, people, but maybe not, maybe gerbils people. I don't know if you can tell it, but the number is 3,000 to 4,000 years ago. That's sort of what you're saying, Rich,
That's
right?
sort of what we're saying.
Yeah.
That's sort of what we're saying.
All right. Now, how do I get
once again, the importance of this paper is that it puts a hard piece of data at least 600. One more little tidbit from the Hopkins paper that caught my attention, because he has a history of, within his history, he talks about people's reaction to the disease, in particular, worshiping various gods, who were to protect them from the disease. So there was in India, a goddess named Shitala Mata, the cool one, Indian goddess of smallpox, apparently the oldest of the deities associated specifically with smallpox, possibly dating as long as 2,000 to 3,000 years ago. Now that's interesting. Usually depicted as riding side saddle on an ass with a broom in one hand and a jug of water in the other. My favorite legend of this goddess origin describes how she flew into such a rage on seeing how ugly she was when she contracted smallpox that she went up into the sky and devoured half the body of the moon, thus explaining its periodic crescent shape. When the chief god, her father, saw this, he said, "I will give you all humanity to eat, but leave the moon alone." After that, she attacked men instead and gave them the smallpox. Worship of this goddess and refusal of vaccination for that reason were still quite strong in parts of northern India during the eradication campaign there in the early 1970s.
There's, if we get her image there.
It's amazing what people come up with, huh?
So that is the most complex of all of religious exemptions.
Yes. Well, there's also a Saint Nicaise, Catholic patron saint of smallpox.
There's also the red treatment, which was just having red things in the room, red furnishings, all this kind of stuff that I'd never heard of.
Yeah.
It's amazing.
I didn't hear that either. Yeah. So the other thing that we talked about a long time ago, Rich, there's a former smallpox hospital on an island in the East River in New York City, which was not originally a smallpox, but it was converted to a smallpox hospital during one of the outbreaks in New York. And they put patients there, and now it's completely ruinous. The windows are all gone. It's falling apart. And
can you visit that?
You can take students on a tour, because I've done it a couple times.
Cool.
What do they have people showing you around?
They do.
And what
they do a whole tour of Roosevelt Island.
Roosevelt Island.
And what do you see in this building? What is it called? Do you know the exact name of it?
I don't know. I've always called it the abandoned smallpox hospital, but there's also a former microbiology lab nearby where a lot of early tuberculosis work was done. And it's just sort of a nice little walk around the island.
Yeah, the Wikipedia page calls it the Smallpox Memorial Hospital.
Yeah. Yes, I actually have taken quite a few photos there. And we also learned during one of the field trips that they filmed a Spider-Man movie there as just a spooky looking place.
Oh, I have to go. I have to go see this.
New York City has a long history of quarantining populations on islands around the main island. There's Hoffman and Swinburne Island down further in the harbor were also used for that for other diseases.
And it seems like Roosevelt Island has been used.
Roosevelt
for that
Island was a very popular, was Roosevelt Island where Typhoid Mary was locked up?
I believe
for a while?
So, yes.
Yeah.
Wow.
Now that we're digressing, I'll bring up two things that I wanted to mention. One that I always like to mention to my students, and that is that just because a virus has pox in the name or disease has pox in the name does not mean it's one of these pox viruses. So we've talked today about the gerbil virus, the cowpox, smallpox, monkeypox, camelpox. Those are all pox viruses. But the chickenpox virus
is not.
Is a herpes virus. And what they have in common is that they make these lesions that are pock-like.
Yes.
And when Rich was talking about this earlier, trying to go back in history and describing the disease, if somebody just had a rash, there was the possibility that it was measles.
Yes.
And so that complicates diagnosing things historically. And then when Vincent read the part about the various targets for eradication of human diseases, that article did not mention the one animal disease that has been
Right.
eradicated, and that's rinderpest, which is related to measles virus. But I wanted to look up the progress on the guinea worm, which is a focus of former President Jimmy Carter and his wife Rosalind and the Carter Center. And this is some pretty amazing statistics. In 1986, when they started the international campaign to eradicate guinea worm disease, there were an estimated 3.5 million cases. And that's been reduced by more than 99.99%. In 2020, there were 27 human cases worldwide. And from January 1st to August 31st of this year, 2021, 10 cases. And those numbers are only provisional. They've not yet been certified. So they're getting really close
They're getting
to
there.
eradicating guinea worm.
That's quite an accomplishment. That's great.
Yeah.
Yeah.
Just adding on to a couple of things you said there. During the smallpox eradication campaign, one of the things that was commonly circulated was a smallpox identification card. And there is a version of that shows the disease. There's a version of that, or several versions of that, that contrast it with the presentation of chickenpox, because chickenpox and smallpox were often misdiagnosed for each other. And so that plays into these historical records. That's one of the reasons you can't necessarily trust a historical record that describes a disease, as you said. The other thing is that the reason it's called smallpox is
to
that
contrast it with the great pox, right?
the great pox, which was syphilis, which is also a blistering disease, but they're big, ugly blisters.
So yours has also been targeted for eradication by WHO. 2012, they said we'll eradicate it by 2020. In 1953, there were between 50 and 150 million cases of yaws globally. Now they missed the eradication. They have, WHO has 153 official cases in 2020 in Papua New Guinea, Solomon Islands, and Ghana. And listen to this. The estimated cost of elimination is $175 million. Like
Peanuts.
Nothing, right?
Wow.
And I remember D.A. Henderson saying, we had hardly anything to eradicate smallpox compared to what's being spent on polio eradication now. It's really remarkable.
What they had was a group of enormously dedicated people, real will, and on a, as is highlighted in that Hopkins paper, on a global scale, people cooperating internationally. He comments in that paper that it was the defining event of his life, and the associations and experiences that he made and had there have persisted throughout the rest of his life. I can imagine.
I do want to point out, though, that level of dedication and that kind of a global network is also present in the polio eradication campaign. There are probably Rotarians listening to this show who are now saying, what does he mean the smallpox people were dedicated? And the Rotarians in particular have been just amazing. And in many places, they're not necessarily doing the whole thing, but there are many other volunteer groups that have gotten involved. And there's this huge network of people who are really, really dedicated to eliminating polio, but there are fundamental problems with the campaign that are hard to overcome.
All right.
Well, that's it, I think.
Thank you, Rich. Very nice.
Sure.
Love it.
Nice.
Good stuff. Do a couple of emails here. I want to say happy birthday to Vivian.
Yes.
Apparently a TWiV fan, has watched every episode since the pandemic began, often 3 to 4 a day to catch up with older episodes. So today is her birthday, Tuesday, October 26th. So happy birthday.
Viviane Bunin, right?
Viviane, yes. And she's from Brazil. She's a physician. And her husband is a former ESPN sports anchor. And two of his brothers went to Columbia. So
Cool.
that's cool. Anyway, happy birthday. Always happy to say happy birthday to people.
Happy birthday. And I apologize that the show notes kind of jumped while you were doing
okay.
that.
No worries.
I put in a couple of photos from trips that I've taken to
Yes.
the smallpox.
That's
hospital
cool.
in Roosevelt Island.
Very cool. So they don't let you go in, obviously, right?
No.
Well, it's
So
falling down.
It's falling down. Yes, that
Yeah.
would be a problem. Too bad. Someone asked me the other night whether I thought there might be infectious pathogens in this old place. I think probably not. It's been a long time since anybody
Lyme
was in
disease.
there.
It's probably full of it.
You think? Yeah, Lyme. Yeah, but not probably not smallpox, right?
Some seasons there's probably West Nile virus you could pick up in there.
West Nile, but not smallpox virus. Nothing
No,
here
no.
that you wouldn't find anywhere else. Yeah.
Exactly.
All right, let's do
And
one.
if you go in with somebody else, then
Yeah,
whatever
of
they're
course.
carrying.
But you're being literal now. Yes.
Yes, I am.
It's okay. Let's do one round of email, and let us start with Rich, please.
Catherine.
Yeah.
Catherine writes, greetings, TWiV. I'm a dentist and fully vaccinated with Moderna since January 2021. I recently developed a headache, sore throat, slight congestion. On day 2 of symptoms, I decided to take an at-home rapid antigen test before going to work. My home test showed up with a clearly visible but not strong positive result. I took a second test in an attempt to confirm the result, which showed a very faint second line. I took a third test, which showed a negative. All 3 tests were taken within 2 hours of each other. I canceled my patients for the day and was able to get a PCR test that afternoon at a site that could provide results while I waited. I was shocked when my PCR test came back negative. I was told I could trust PCR results. My question is, what is the shortest amount of time someone could have a COVID infection that's strong enough to produce a positive test result? Is it possible that someone could only test positive for a day or 2 before their immune system clears the virus? Thanks for your thoughts. P.S. I thought I would also share that I live in northern Michigan. The nearest site that could give me a same-day PCR test was an hour and a half away. For what it's worth, it was worth it to me to drive that distance and avoid all the personal hassle of waiting 4 days, the norm in this area, not knowing if my 2 young, unvaccinated, ineligible children were at risk or needing to quarantine them unnecessarily, and having to cancel days of patients while I awaited the results. This still seems a travesty to me that timely PCR results are that difficult to access. Appreciate your show. Thanks for all your discussions and insights. Okay. What's the shortest window of time that you could show up? Well, first of all, it depends on the test. Okay. Because the PCR test is going to be positive if you look at the time course of the infection. My sort of picture is that the PCR test is going to turn positive before the antigen test. And then the antigen test is going to turn negative, and you're still going to have positive PCR out the other end. Okay. And I don't know, I would say that a rapid antigen test, that's going to show up for what, 5 days, a week, longer than that, or certainly PCR test, probably 2 weeks. Okay. But if you, if you're thinking that you might have tested positive with a rapid antigen test in the morning and negative with a PCR test in the afternoon, I don't think so.
No.
Sounds like the rapid antigen test was lying to you. But it's weird.
Sounds like it was
2 out of
negative,
3 would give you a soft lie.
Yeah.
So I don't have an explanation for that. Anybody else?
No, but the third time was negative. So I'm guessing it's just a borderline false positive. If it's so weak, right? I don't know. But I think you're right. You're not going to go from positive to negative in a day. Yeah.
And you did all the right stuff.
Yeah.
Cancel all your appointments. Get that PCR test as fast as you can.
I agree that.
And-
I'm sorry. Go ahead, Kathy. Go ahead.
I was just going to say, I agree that it seems a travesty that you have to
Yes.
drive an hour and a
Yeah.
half,
Yeah.
or else wait for 4 days. We're pretty lucky here in Ann Arbor. We can go get a saliva PCR test and get the result pretty much within 12 hours,
Yeah.
same day.
Yeah. I mean, that would be. It really should be all over that it's easy to get. It is a travesty. Yep.
Yeah, you should be able to FedEx the swab in. I mean, next day would be the appropriate timeline for that. I'm sure that can be done, but
Yeah. Kathy, can you take the next one?
Helen writes, "Hi, I listen to your shows all the time and really enjoy them. Microbe TV performs a great public service. I thought you might like to hear about my dad, and I have some questions about COVID. My father is 97 years old, has no chronic health conditions, walks a mile every day, and lives alone in Mississippi. He got his second Moderna vaccine for COVID February 25th. Prior to getting his third vaccination, his doctor required him to get his antibodies tested, which he did on August 31st. The test result was 0. I'm not sure what they tested, but he was told he could get his third shot, and he got it on September 3rd. On October 19th, he got his antibodies tested at his doctor's office, and the result was, quote, 55. The technician told my father that this was a, quote, good result. My father is now thinking that he may have a shield against COVID, and he should be safe to start socializing more, going to church, supper club, Lions Club, and the YMCA, activities that he has not been able to enjoy for over a year. He's not very happy about wearing his face mask, but he does when he occasionally has to go someplace like the drugstore. I'm not sure how much he would wear it when he socializes. So my questions are, what should I think about all this? Can you help me understand the result of 55? I'm sorry I don't have more information about the test. My father is of the generation that felt like asking a doctor too many questions might insult them by questioning their competence. What is the usual duration of antibodies? Or what could be expected for someone his age? How long of a window of time might it be relatively safe for him to socialize more? Or do antibodies have anything to do with this? I'm not an immunologist or a virologist and would really appreciate it if you could take a few minutes to explain a bit how the immune system works. I hear you saying that antibodies are not the whole story and there is other immunity such as memory B cells, ? I would love to see you draw a picture to explain a little about all these cells. If memory cells are important, would it be helpful for them to be measured? I know I've rambled on a lot, but I would love to hear your feedback about my dad's situation. By the way, I'm in North Carolina where the weather is getting cool at night and my cherry tomato plants are still making flowers, which I am hoping will have time before frost to develop into fruit. Thanks for your response. Helen. So I think we've heard several times from Daniel, and Vincent has reiterated it, that we really can't trust numbers from antibody test results. So the test result that was 0, not sure what that means. I also have no idea what a result of 55 means. And, the technician telling him that's a good result, we don't know what that means. I think Daniel has come out pretty strongly that, doing antibody tests and making decisions based on that is, we don't have the data for that at the moment. So whether he's safe to start socializing more, doing all those things, I would say he's 97 years old. If he wants to do those things, and if he can wear a mask to do most of those things, he should do them. Yeah. That would be my advice. He's been vaccinated, wear a mask, and go do those things that are socializing and important. In terms of the questions of how long of a window of time might it be relatively safe for him to socialize more? I think, if he's now had 3 vaccinations with Moderna, he should be good to go for a good length of time. But again, we don't really have the data on how long all these responses will last. There is more to the story than just antibodies. And if we want to have the brief explanation of what other things come into play, I'm willing to defer to someone else to do that.
Maybe
Brianne, take it away.
Maybe she should. Maybe she should.
30
pick
seconds.
Up,
Explain the immune system.
Brianne,
Go.
maybe she should pick up that book by Sompirak, How the Immune System Works.
Mm-hmm.
Yes.
That has nice
yes.
little pictures of cells, right?
It does have nice little pictures of cells. And the brief answer, Helen, is that antibodies are sort of tools that are made by memory B cells. So really you care about how many memory B cells you have to make more tools when you need them. Similarly, there's some memory T cells that can be important here. It would be great if you could get them measured, but we don't have a really good way to have them measured at your doctor's office easily. And I would say that I agree with everything that Kathy said. I don't know 55 what. But if it were my family member, I would say a lot of the same things. I would just ask them to think about how much virus there is circulating in their community as one other factor.
Yeah, I agree.
Yeah, and just to reiterate, 97 years old, has been away from all of his social activities for a year. He's now triple vaccinated. His odds are as good as they're going to get against this virus. And hopefully he gets his flu shot too, because that's important, especially in his age group. Probably at this point, a bigger risk than SARS-CoV-2 now that he's fully vaccinated against the latter. And yeah, you don't live forever. He should go out and enjoy himself.
Helen, your dad is my hero.
Yes. Yes, we aspire to be him.
And I agree with everything that's been said, but it's a tough question. I mean, clearly, he got what we would call in the smallpox vaccination world, he got a take. Okay, he got an immune response, and that's about all you can say. Okay, and you don't really know how good that is. I agree with Alan that you're not going to get much better than that, and the risk is never going to be zero. And so you need to take the other factors into account. Are you hanging out with unvaccinated people? Are you hanging out with vaccinated people? Are people wearing masks? Are you indoors? Are you outdoors?
Are you wearing your seatbelt?
Yeah. What's the virus load in your community? That's an important one. Okay. And so try and factor all those things in. And as Daniel says, stay safe. Okay. But at this point, I don't think staying home makes any sense.
And I will point out, in terms of having or not having the data, there was a preprint that came out yesterday that's on my list to read about the Moderna vaccine challenge in primates a year after vaccination.
Yeah. Right.
So just to reiterate, yes, antibodies aren't the full story. They're produced by B cells, but there's also immunity that's conferred by another type of cell, the T cells, and that's what we can't really measure.
Right.
So,
and you should go to Amazon and buy How the Immune System Works by Lauren Sompirak. That's a nice small book with nice illustrations that would probably go a long way to explaining about these cells. Since we're mostly an audio show, it would be better for you to get that. Alan, sorry, go ahead.
There's another book that's coming out soon that I've seen information about. I have pre-ordered it and I am supposed to have it on November 2nd. So perhaps I will have another recommendation for people soon.
All right. Alan, can you take the next one?
Sure. Charles writes, hello, Twivers. A great day in North Carolina, 71 Fahrenheit, 22 C, low humidity, and just a few clouds. If it was like this all year round, central North Carolina would have a population of around 100 million. During TWiV 818, part of the discussion made me want to ask what traits good scientists share. Praying to Google brought up a bunch of good traits, but missed a couple of very important points. First, good scientists have to have almost no fear of being wrong. Not that scientists want to be wrong, but that their egos don't take a devastating hit when they are wrong, and they see it as an opportunity. A scientist that is never wrong has never been right about anything that is important or is very young. Second, most of the lists of traits talked about failed experiments without knowing what a failed experiment is. To the layperson, a failed experiment is one that did not turn out how the scientist wanted or had predicted. In reality, a failed experiment is one that failed to answer the question, not one that failed to give the expected answer. Here's an example of a failed experiment of which I have firsthand knowledge. After hours of setup and preliminary data collection, a frequency generator was set to 60 hertz, not 60 cycles per minute. Bad stuff happened. To make matters worse, we looked in the wrong place for the problem, so it happened again. After the second failure, we did find the problem and added a second check of the frequency generator to the procedures for the experiment. At about $10,000 per run, those were expensive mistakes. Thanks, Charles. P.S. Is Tom Lehrer going to be anybody's pick? Possibly one of Dixon's, I'm not sure. Yeah, that's a very good point, that expecting to be wrong a lot and understanding what a failed experiment is. A failed experiment didn't give you information. You know, if it gave you information, then the experiment worked. If the information was not what you were expecting, okay, that's science.
Yeah, that's not failed though, right? Yeah.
No, that's not failed.
It answered the question, yeah. Then there's a failed experiment because you set it up wrong, which he's talking about here, and that's different, right? Yeah.
Yeah. Yeah. One of the things I always think about is, you know, when you set up an experiment, there's your sort of experimental protocol, and then there's many, many more controls. Okay. Because as you set it up, you try and think of all of the ways that you could be wrong. Okay. And all of the sort of information that you could get that would be, you know, wrong. And you try and control for all that. Okay. So that once the experimental protocol is confined by all those controls, it can't be anything else. All right. And invariably you do the experiment and the answer is, hey, did you think of this? Right. There's a control you didn't do. Right. Or two. So you do it.
There's a I think it was a PhD comics where the fellow goes to his advisor, you know, very advisorial looking guy with a beard, and says that we just had an accident in the lab. And the advisor replies, "Oh my gosh, was anybody hurt?" "No." "Are we going to be sued?" "I don't think so." "Well, did you clean it up?" "Yes." "Did it produce good data?" "Yes, actually it did." "Can you do it again?" So, yeah.
I don't know. I think you early on on a path to being a scientist, you find out about failure, and people who can't handle it leave. Yeah, I know many graduate students after a few months say, "This is not for me," and leave. And part of it is failure. Yeah, and those of us who stick around can deal with it. It's fine.
That's why, as with many professions, when you interview candidates, you ask them about their lab experience, and you're not asking to find out, you know, all the cool things they did. You're asking to find out whether they have enough experience to know what they're getting into, which is a lot of failure.
All right, one more from Diana. Just in case you did not notice, Merck published a response, and the authors followed with a response to Merck to the Zhao et al. Paper on genetic toxicity in mammalian cell culture with the molnupiravir metabolite TWiV-818. Thanks for all the TWiVs, immunes, TWiPs, and TWIMs. All right, so Diana sent the Merck, it's called Letter to the Editor in response to Zhao et al. This is from people at Merck and also Amory, who were involved in the early development of this drug. And that was many years ago, 5 years ago or something like that. The early name of the drug was EIDD some number, and it's Emory Institute of Drug Development. And so basically they say here that this drug has been examined way more than you guys did and in many more assays. Yeah. And we didn't find any genotoxicity. All of this provided what they call strong evidence of lack of in vivo relevance to your results, basically.
Right. They did in vivo models, two
Yes.
distinct rodent mutagenicity in vivo models.
Yeah. And they say while molnupiravir and or NHC have demonstrated the ability to induce mutations under specific in vitro conditions, including the ones that they published, and also an Ames test, it's positive in an Ames test. In animals, there's no genotoxicity. And they say that's the bottom line. The animal test is really what you need to do, and they detail what they did. And then you say, your assay is not really what we're interested in. It's a nice assay, but it's not standardized. It's not acceptable in pharmaceutical industry and so forth. You did things that were a little suspect, and 32 days of exposure and so forth. Yeah. They're not standard assays. All right, so they put a whole bunch of bullet points. And then the authors, of course, in turn reply, and they say, thank you very much, which is what you always do.
Yes.
and they say, we view our work as providing the proof of concept, showing that it can be converted to a DNA and DNTP and become a mutagen. So it's possible that this could happen. You have to look over long periods of time. They kind of defend their assay, but
their experimental design.
Yeah, their design. They explain some of the details that the Merck article brought up. So, I think in the end, what we have to see is in the long term, is this going to have any effect? I think the assays, there's a history of doing these animal tox assays and putting drugs into people. They correlate. That's why we still do them. So I don't know. Any thoughts, ladies and gentlemen?
The line at the end where they say, it's hard to argue that a ribonucleoside precursor to both RNA and DNA goes into one, but not the other. And so I think that's kind of the point they were trying to make, is that this thing is a precursor for RNA, but it's also a precursor for DNA, and then that could have results on DNA replication and repair.
Right. But then the Merck people would say, but we looked in animals and it doesn't cause cancer. It doesn't cause genotoxicity anyway. So
I think, we can make everybody feel happy and say, yeah, it's a proof of concept. But if you do the appropriate experiments for the FDA, the concept doesn't play out in a real world in vivo situation.
Yeah, you always do a proof of concept and then next steps.
Yes.
In these drugs, you do an Ames test first, and if it's positive, then you go on to the animals, and then if those are positive, then you don't put the drug in people. But if they're negative, you do. I just don't see. Basically, they did an Ames sort of test in eukaryotic cells, right? So that would be the first thing you do. Then you do the animal test, and they're negative. So the drug can become a DNA base, and it can insert into DNA, but apparently it doesn't happen in an animal. So I'm okay.
Or at least not at a frequency you need to worry about.
Yeah. Okay. Let's do some picks. Brianne, what do you have for us?
So I have something called The Animal Kingdom is Full of Genetic Screwballs. And I read about this. I read this article, and it was another example of me thinking, how am I a biology professor who's never heard of this? Why did no one tell me? And so this is actually talking about a phenomenon that happens in a number of different organisms, some of which are some parasites that Dixon would know a bit about. Also happens in some birds and some others, where there are chromosomes that are thrown out of all of the cells that are not germ cells. And so those chromosomes seem to go from germ cell to germ cell, go across the generations. But then as the embryo develops and all of the other cell types differentiate, they throw out or get rid of some of the other chromosomes. And so it talks about that and sort of the idea that for a long time, people thought that those actually didn't encode any genes, but in fact they do. And those genes are quite active in germ cells. And just some of the studies of this and exactly how much DNA may be involved, whether we're talking about gnats or lampreys or some particular roundworms or things like that. In some cases, this is 10% of the genome or more. And I just thought this was absolutely fascinating to realize that there are some processes happening with DNA that sort of go against our sort of canonical understanding and that are just really cool and that we need to learn more about.
And that this is widespread.
Right.
This is not just one obscure species that does this weird thing. It's like apparently all songbirds may actually have something like this going on with their genome.
Yeah, it's crazy.
Yeah.
So do all the cells in a given organism throw out the same set of chromosomes?
It looks like it.
Okay.
That is my understanding. Yes.
Yeah.
Right.
And they also point out here, which was something that I had thought about, is, well, we know lots of situations where you might silence genes that you don't want to turn on. But this is sort of extreme. Why do you need to do this instead of silencing the genes?
Well, it sure takes care of the problem.
Yeah. Are silenced.
And it does raise another problem, an organizational problem. You got to put all the genes that you want to get rid of in one place.
Yes.
Yes.
So you can get rid of them.
Yep. Then what?
Interesting.
Pox virus puts them at the ends, right?
That's true. And actually, I was thinking about this today because, hey, Kathy, is it not true that adenoviruses have sort of more variability at the ends of the genome than in the middle? No? Okay.
No, it's that they have their important stuff kind of at the ends. The E1A is always at one end, and something like E4 is at the other end, and then right in from E1A is from E1 is the E2 replication stuff.
All right.
Brianne, the answer to your rhetorical question, why didn't anyone ever tell me, is because you need to find out on your own.
That's true. That's true.
You're going to learn it all on your own. Nobody's going to tell you. Yeah.
I know, but I just thought that this was, a fascinating piece of biology that's out there that I didn't know about.
Yeah. Oh, here on TWiV, you find out about it because we share things, right? Kathy, what do you have for us?
First, I want to mention that we have more vaccine town halls on the schedule, posted more Zoom links. So go to asv.org/education with vaccines soon probably coming into play for younger kids and boosters from Moderna and J&J and so forth. You or your friends or relatives may have questions. So asv.org/education. I picked a site from Svaha, SVAHA, which has a lot of science dresses and accessories. I found out about this because a friend of mine ordered one of the dresses that's made from fabric that has Rosalind Franklin's DNA diffraction image on it. And so she sent me that image, and then when I asked her for the link, she gave me the link. And then for her particular dress, they're in short supply, and she said, oh yeah, I had to order one that was 2XL, and then I just basically took it all apart and remade it to fit me. So you may not want to get your heart set on that particular dress, but there's a lot of different science things, physics, biology, engineering, all different kinds of links if you go to this site. And this was started by a person who has a PhD in industry apparel, and she recognized a need for science and tech-themed clothes for girls because she couldn't find any astronaut shirts for her daughter.
Oh.
So they also have mugs and tote bags, just all kinds of things, some jewelry. So I think it's sort of a science, art, accessories thing that we haven't seen before, but I thought it was pretty cool.
Very neat.
Brianne, did you see the antibody necklace?
That was what I was just noticing.
It's cool. Very cool.
Excellent.
Yeah. But they run, that one at least runs small, huh?
Yeah.
She had to take it apart.
Well, her dress was 2XL. No, it was very large, and she had to cut it down. Because I wrote to her and said, did you buy just the fabric or did you buy the dress?
Oh, I see.
She said, well, sort of. I bought the fabric because I had to take it apart. And
I misunderstood because she's not a large person.
No, right. She bought a very large dress and cut it down and made it fit.
Rich, what do you...
I've got lots of things I want.
Yeah, it's cool. Rich, what do you have for us?
I have a pick that is definitely not science, but definitely appropriate to the episode. It is a series of historical novels that is called The Saxon Stories. The author is Bernard Cornwell. And this is basically around the time of Alfred the Great, 9th, 10th century CE, when during the period of time that the Danes and the Saxons were duking it out in the British Isles. Okay. And Cornwell has crafted a main character who speaks in the first person cleverly as a Saxon who, as a young boy, was in the heat of a battle kidnapped by the Danes and raised with the Danes. Okay. And then ultimately, through a variety of circumstances, winds up he's a warrior, winds up fighting for Alfred. So he has insights into both sides of this conflict, okay, over a 5-6 decades period of time. I'm on book 5, and it is it's well, I haven't seen much of Game of Thrones, but it's blood and gore and guts and veins in your teeth for sure. Okay, it's good stuff.
Cool.
So this is like Ethelred the Unready, right?
Yeah,
same idea.
Exactly.
There are probably characters in the background who are getting smallpox.
Right.
Cool. Alan, what do you have for us?
I've had this in my list of picks for a few weeks. I decided I wanted to pick it right before Halloween. So that's when this episode is going out. This is a little bit of medical history. It's a reprint of a paper that summarizes the history of one local practitioner of a profession called resurrectionist. Now, a resurrectionist was somebody, a contractor, freelancer, if you will, who in the early 1800s would go around supplying cadavers to medical schools. Because at the time, medical schools had no legal means of obtaining cadavers. So the typical arrangement was that the head of the anatomy department would tell the janitor, hey, we need cadavers. And the janitor was supposed to sort it out. So there was plausible deniability, and you'll understand in a moment why we needed that. The janitor would put word out that he needed cadavers, and people who were enterprising would go out to graveyards the night after a funeral and dig some up and take them over to the anatomy lab and deliver them through the back door to the janitor. And hey, what do you know? Cadavers showed up. Isn't that nice? So this details the colorful history of one particular resurrectionist who operated in the Cincinnati area. His last name was Cunningham, and he became known as Old Cunny because he was also very cunning in getting and keeping these corpses to get to the medical school. And it's a series of interesting and amusing stories. If you liked Weekend at Bernie's, you'll probably get a kick out of some of these. Some of his strategies, and I found it interesting enough that I did it as a free audiobook with LibriVox, which is this public domain audiobook thing that I do on occasion. And so I've also linked to their short nonfiction collection, volume 84, and it's the third piece in that. So this is it's a reprint of an article, take you about 15 minutes to read, or if you want to take about 20 minutes and hear me read it, you can do that. That's an alternative.
Oh, so you read
Halloween.
it. You read it.
I read it.
Oh,
yes.
Cool. Very good. Yeah, under the subject, one of the topics is body snatching.
Yes.
There are no more cadavers, right? It's all done on computers now, right?
Now it's, but they got so this piece ends with, I think in the 1850s, they finally, legislatures were persuaded to pass laws that provided a legitimate conduit for people to donate bodies to medical schools. And so resurrectionists were put out of business, and people like Old Cunny no longer had to ply their trade with picks and shovels.
Right. Cool. All right. My this is installment number 4 in Seminal Papers in Molecular Biology. This is a 1978 Nature paper by Taniguchi, Palmieri, and Weissman. Q-beta DNA containing hybrid plasmids giving rise to Q-beta phage formation in the bacterial host. And I think I might be the only person who would ever pick this as a seminal paper. It's not one of the ones you'd think about, but I think everyone these days, of course, uses infectious DNA copies of viral genomes to do all sorts of manipulations. And this is really the first one for a virus. I think it's an RNA virus. Q beta is a bacteriophage of E. coli. It's a plus-stranded RNA bacteriophage used in many laboratories as a wonderful model organism. It reproduces very quickly, and you can get large quantities of virus. You can get lots of plus and minus stranded RNA. And so what they did here was to make a DNA copy using reverse transcriptase. So this is now late 70s. We are able to copy RNAs with RT and clone them into bacterial plasmids. And they make RNA transcripts in the bacteria themselves. And they don't do in vitro transcription. They do. They put the plasmid in the bacterium, and out comes bacteriophages. And it's a bit quaint because they say the availability of a plasmid that would have a DNA copy of Q beta would greatly facilitate the preparation and study of unconditionally lethal mutants and would allow genetic manipulations that cannot be carried out at the RNA level, right? So that's all true. But I think the focus on lethal mutants is pretty funny. So this is the first time this was done for a virus. And I think it's important because it led to everything else. I was certainly aware of it when I made infectious polio RNA, I don't know, 3-4 years later than this. I looked at this paper quite a bit, although here they put the plasmid right into bacteria. And then there's an interesting discussion about how does this work? How does a DNA plasmid initiate an infectious cycle for an RNA virus? And they say, there must be a sequence somewhere that's making RNA. It can't be initiating at just the right place for the virus. How would that work? It has to be probably trimmed a little bit. These are all the same things we were thinking about many years later with polio virus. So I think it's a seminal paper. But most people who make infectious clones probably don't even know about it, right?
Charles Weissman celebrated his 90th birthday just 12 days ago.
Wow. Good. We have a couple of listener picks. One of them is contributed by Kathy. Why don't you tell us about that? You're muted.
Sorry, evidently this was this was kind of all over the interwebs, social media and stuff. It's an article called "No Time to Die: An In-Depth Analysis of James Bond's Exposure to Infectious Agents," and pretty much the title says it all. It's published in Travel Medicine and Infectious Disease.
That's cool.
So,
It is hilarious.
This
The
is
abstract
great.
in particular. The
Yes.
abstract is priceless.
yes.
Huh. I haven't looked at this. I'll look at this.
That's great.
Global travelers, whether tourists or secret agents, are exposed to a smorgasbord of infectious agents. We hypothesize that agents preoccupied with espionage and counterterrorism may, at their peril, fail to correctly prioritize travel medicine.
Yeah, it mentions like how many times he's seen ever washing his hands and things like that.
We uncovered above average sexual activity, often without sufficient time for an exchange of sexual history, with a remarkably high mortality among Bond's sexual partners.
That's funny. That's funny. So
thanks,
I
Justin.
actually have a book I picked years ago, which is kind of a similar thing. It's a summary of all the infections that killed kings, right? I think it's not just British, it's kings throughout Europe, and they go through, it's hard to know what a lot of these people died of, right? But some of them are clearly infectious diseases. It's called The Death of Kings. Kind of ties in with our episode today. We have also a pick from Jeff. My dear Twivers, in my humble opinion, Tufeki, the author of this Times article, does a good job in segmenting the, quotes, unvaccinated into several useful classifications based on a decent amount of data, along with some extrapolation. Since this topic arises almost every other show, it could prove useful. And this is an opinion piece. The unvaccinated may not be who you think. And I haven't logged in, so I can't actually see it. Yours in scientifically based skepticism, Jeff, only a computer scientist and lifelong learner. All right. Thank you, Jeff.
I have not read this, but I presume this is, we have a tendency to lump the all unvaccinated all into one bunch. And it needs to be acknowledged that there are many, many different reasons why people choose not to be vaccinated, all of which have to be taken into account, in particular when we're trying to peddle vaccine education.
Or not even necessarily choose, but simply haven't been vaccinated yet.
Yes.
Haven't been. Yes, right. Right.
I agree. I read this when it came out, and I was just now reaching around on my desk because I was pretty sure I ripped it out, but it's probably on my pile of articles at home. Some of them become worthy of bringing in here, but yeah.
That's TWiV 822. The show notes are at microbe.tv/TWiV. If you want to send a question or comment or pick of the week, TWiV at microbe.tv. If you'd like to support what we do, microbe.tv/contribute. Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Looking forward to part 2 of Poxy Stories.
Yeah.
Was that going to be like a year from now?
I don't know. I asked for I have a collaborator in this.
Yes.
Okay.
Right.
We will have a guest at the time.
Good.
And so I don't know quite when it's going to be.
All right. Kathy Spindler, University of Michigan, Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
Alan Dove is at alandove.com. Alan Dove on Twitter. Thanks, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.